students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामची प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक यू. 21 Day Biology Reading Challenge Day 9 Reading Challenge Chapter number 9 from the NCERT Biology textbook of 11th Standard. Name of chapter is the Biomolecules. There is a wide diversity in the living organism in our biosphere. Now a question that arises in our mind is, are all living organisms are made up of the same chemicals, that is the elements and the compounds? You have learned in the chemistry how element analysis is performed. If we perform such an analysis on the plant tissues, animal tissues or a microbial paste, we obtain a list of the chemicals like the carbohydrates, hydrogen, oxygen and the several other their respective contents per unit mass of the living tissues. If the same analysis is performed on the piece of the earth crust is an example of the non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute term, no such differences could be made out. All the elements present in the sample of the earth crust are also present in the sample of the living tissues. However, a closer examination reveals that the relative abundance of the carbon and the hydrogen with respect to their element is higher in any living organism than in the earth crust. 9.1 How to analyze the chemical composition. We can continue the asking in the same way. What type of the organic compound are found in the living organisms? How do one go out the finding the answers? To get an answer, one has to perform a chemical analysis. We can take the any living tissue like a vegetable or a piece of the liver, etc. and grind it in the trichloroacetic acid using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through the cheesecloth or a muslin cloth or a cotton, we would obtain a two fractions. One is called as a filtrate or more technically the acid soluble pool, and the second is the retentate or the acid insoluble fraction. Scientists have found the thousands of the organic compound in the acid soluble pools. In higher classes we will learn about the how to analyze the living tissue sample and identify a particular organic compounds. It will suffice to say here that one extract the compounds, then subjects the extract to various separation technique till one has separated a molecule or compound from all other compounds. In other words, one isolate and purify a compound. Analytical techniques, when applied to the compound, given us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from the living tissue can be called as a biomolecules. However, the living organisms have also got a inorganic elements and the compound in them. How do we know this? A slightly different but a destructive experiment has to be done. One weight a small amount of the living tissue, say a leaf or a liver, and this is called as a weight, and dry it. All the water get evaporate, the remaining material given a dry weight. Now if the tissue is fully burned, all the carbon compounds are oxidized to the gaseous form like the CO2 and the water vapors and are removed. What is remaining is called as ash. This ash contains inorganic elements like the calcium, magnesium, etc. Inorganic compounds like the sulfate, phosphorus, etc. are also seen in the acid soluble fraction. Therefore, the elemental analysis gives the elemental composition of the living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While the analysis for the compound given and the idea of the kind of the organic and the inorganic constituent present in the living tissues. From the chemistry point of view, one can identify the functional group of the aldehyde, ketones, aromatic compounds, etc. But from the biological point of view, we shall classify them into the amino acids, nucleic bases, nucleotide bases, then the fatty acid, etc. Amino acid are the organic compound containing and the amino group and the acidic group is the constitute of the same carbon, that is the alpha carbon. Hence they are called as a alpha amino acid. They are substituted the methane. There are the 4 substituent groups occupy the 4 valency position, and these are the hydrogen, carboxyl group, amino group, and the various groups designated as a R group. Based on the nature of the R group, there are the many amino acids. However, those which occur in proteins are only of the 20 types. The R groups in this proteinaceous amino acid could be the hydrogen. The amino acid is called as a glycine, a methyl group, that is the alanine, then the hydroxymethyl, that is the serine, etc. 3 of the 20 are shown in the figure. The chemical and the physical property of the amino acid are essentially of the amino acid, carboxyl, and the R functional group. Based on the number of amino acid and the carboxyl group, there are acids. For example, the glutamic acid. Basic are the lysine and the neutral are the valine amino acids. Similarly, there are aromatic amino acids like the tyrosine, phenylalanine, tryptophan. A particular property of the amino acid is the ionizable nature of the NH2 and the COOH group. Hence, in a solution of the different pH, the structure of amino acid get changed. Lipids are generally the water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to and the R group. The R group could be the methyl, that is the CH3, or ethyl, that is the C2H2, and the higher number of the CH2 group, that is the 1 carbon and the 19 carbons. 1 carbon to the 19 carbons. For example, the palmitic acid has the 16 carbon including the carboxylic carbon. Arachidonic acid has the 20 carbon atoms including the carboxyl carbon. Fatty acid could be saturated without double bond or unsaturated with one or the more carbon-carbon double bonds. Another simple lipid is a glycerol, which is a trihydroxypropane. Many lipids have the both glycerol and the fatty acid. Here, the fatty acids are found esterified with the glycerol. They can be then monoglycerides, diglycerides, and the triglycerides. They are also called as a fats and the oil based on the melting points. Oil have a lower melting point, for example, gingerly oils, and hence remain as a oil in a winter. Can you identify a fat from the market? Some lipids have the phosphorus and the phosphorylated organic compound in them. There are the phospholipids. They are found in the cell membrane. Lecithin is the one example. Some tissue, especially the neutral tissues or neural tissues, have a lipid with a more complex structures. Living organisms have a number of carbon compounds in which the heterocyclic rings can be found. Some of these are the nitrogen bases: adenine, guanine, cytosine, uracil, and the thymine. When found attached to the sugar, they are called as the nucleosides. If the phosphate group is found esterified to the sugar, they are called as nucleotides. Adenosine, guanosine, thymine, uridine and the cytidine are the nucleosides. Adenylic acid, thymidilic acid and guanylic acid, uridylic acid and the cytidilic acid are the nucleotides. Nucleic acid like the DNA and the RNA consist of the nucleotides only. DNA and the RNA function as a genetic material. Now the 9.2, the primary and the secondary metabolites. The most exciting aspect of the chemistry deals with the isolating 1,000 of compounds, small and the big, from the living organisms, determining their structures and if possible the synthesizing them. If one were to make the list of the biomolecules, such as the list would have the 1,000 of organic compounds including the amino acid, sugar, etc. For reason that given in a section 9.10, we can call them as a biomolecules as a metabolites. In animal tissue, one notices the presence of all such categories of the compounds shown in the figure 9.1. They are called as a primary metabolites. However, when one analyze the plant, fungal and the microbial cell, one would see the 1,000 of compounds other than these called as a primary metabolite. For example, the alkaloid, flavonoids, rubber, essential oil, antibiotics, color pigment, scents, gums, spices, etc. These are called as a secondary metabolites. While the primary metabolites have the identifiable functions and play a known role in the normal physiological processes, we do not at the moment understand the role or the function of all the secondary metabolite in a host organism. However, the many of them are the useful to the human welfare. For example, the rubber, drugs, spices, scent and the pigment, etc. Some secondary metabolites have the ecological importance. In the latter chapter, the years you will learn more about this. Now the some secondary metabolites are the, for example, pigments like the carotenoid and anthocyanin. Then alkaloids like the morphine and the codeine. Then terpenoids are the monoterpenes and diterpene, etc. Essential oil like the lemon and the grass oil, etc. Then the toxins like the abrine and the ricin. Lectins like the concanavalin A and the drugs like the vinblastine and the curcumin, etc. Polymeric substances like the rubber, gum and the cellulose are present. Now the 9.3 is a biomacromolecules. There are the one feature common to all those compounds found in the acid soluble pool. They have the molecular weight ranging from the 18 to around the 8,000 Daltons approximately. The acid soluble fractions has only the 4 types of the organic compound, that is the protein, nucleic acid, polysaccharides and the lipids. These classes of the compounds, with the exception of the lipid, have the molecular weight in the range of the 10,000 Daltons and above. For this very reason, biomolecules, that is the chemical compound found in the living organisms, are of the 2 types. One, those which have a molecular weight less than the 1000 Dalton and are usually referred to as the micromolecules or simple biomolecules, while those which are found in the acid insoluble fraction are called as a macromolecules or a biomacromolecules. The molecule in the insoluble fraction, with exception of the lipid, are the polymeric substances. Then why do the lipids, whose molecular weight do not exceed the 800 Dalton, come under the acid insoluble fraction, that is the macromolecular fraction? The lipids are indeed a small molecular weight compound and are present not only as such but also arranged into the structure like the cell membrane and the other membranes. When we grind a tissue, we are disrupting a cell structure. A cell membrane and the other membrane are broken into the pieces and forms a vesicles which are not water soluble. Therefore, these membranes fragments in the form of the vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represent roughly the cytoplasmic composition. The macromolecules from the cytoplasm and the organelle become a acid insoluble fraction. Together they represent the entire chemical composition of the living tissue or the organism. In summary, if we represent the chemical composition of the living tissues from abundance point of view and arrange them class-wise, we observe that the water is the most abundant chemical in the living organism. Now the average composition of the cell, the component and the percentage of the total cellular mass, that is the water, is about the 70 to 90%. Proteins are the 10 to 15%. Carbohydrates are the up to the 3%. Lipids are up to 2%. Nucleic acid up to the 5 to 7%, and ions are 1%. Now the 9.4 proteins. Proteins are the polypeptide. They are the linear chain of the amino acid linked by the peptide bond are shown in the figure. Each protein is a polymer of the amino acids, as there are the 20 types of the amino acids. For example, the alanine, cysteine, then the proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. The homopolymer has only one type of the monomer repeating n number of times. This information about the amino acid content is the important, as later in your nutrition lesson you will learn that a certain amino acids are essential for our health and they have to be supplied through our diet. Hence the dietary protein are the source of the essential amino acids. Therefore the amino acid can be essential or the non-essential. The latter are those which our body can make, while we get essential amino acids through our diet or food. The proteins carry out the many functions in the living organism. Some transport nutrients across the cell membrane. Some fight the infectious organism. Some are the hormones. Some are the enzymes, etc. The collagen is the most abundant protein in the animal world, and the ribulose bisphosphate carboxylase oxygenase, that is the rubisco, is the most abundant protein in the world of the biosphere. Now the some proteins and their functions, like the collagen and their function is the intracellular ground substances. Trypsin is the enzyme. Insulin act as an hormone. Antibodies, they fights the infectious agent. Receptor, these are the sensory receptor for the smell, taste and the hormone, etc. Then the GLUT4, it is the enable the glucose transport into the cell. Now the 9.5 it is the polysaccharides. The acid insoluble pellet also has a polysaccharide like the carbohydrate as another class of the macromolecule. Polysaccharides are the long chain of the sugars, and they are the threads or the literally a cotton threads containing a different monosaccharides as a building blocks. For example, the cellulose is a polymeric polysaccharide consisting of only one type of the monosaccharide, that is the glucose. The cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of the energy in the plant tissue. Animals have the another variant called as a glycogen. Inulin is the polymer of the fructose. In the polysaccharide chain says the glycogen, the right end is called as a reducing end and the left end is called as a non-reducing end. It has a branch as shown in the form of the cartoon, and the starch form the helical secondary structures. In fact, a starch can hold a I2 molecules in the helical portions. The starch I2 is a blue in the color, and the cellulose does not contain the complex helix and hence cannot hold the I2. The plant cell wall are made up of the cellulose, and the paper made from the plant pulp and the cotton fiber is a cellulosic. There are the more complex polysaccharides in the nature. They have the building blocks, amino sugars and the chemically modified sugars, for example glucosamine, N-acetyl glucosamine, etc. The exoskeleton of the arthropod, for example, have a complex polysaccharide called as a chitin. These complex polysaccharides are mostly homopolymer. Now the 9.6 is the nucleic acid. The other type of the macromolecule that one would find in the acid insoluble fraction of the any living tissue is the nucleic acid. These are the polynucleotide. Together with the polysaccharide and the polypeptide, these comprises the true macromolecule fraction of any living tissue or the cell. For the nucleic acid, the building block is a nucleotide. The nucleotide has a three chemical distinct compound. One is the heterocyclic compound, the second is the monosaccharide, and the third is the phosphoric acid or the phosphate. As you notice in the figure, the heterocyclic compound in the nucleic acid are the nitrogenous bases named as adenine, guanine, uracil, cytosine, and the thymine. Adenine and the guanine are the substituent purines, while the rest are the substituent pyrimidines. The skeletal heterocyclic ring is called as a purine and the pyrimidine respectively. The sugar found in the polynucleotide are either the ribose, that is the monosaccharide pentose, or the 2'-deoxyribose. A nucleic acid containing the deoxyribose is called as a deoxyribonucleic acid, that is the DNA, while that which contain the ribose is called as a ribonucleic acid, that is the RNA. Now the 9.7 is the structure of the protein. Protein, as mentioned earlier, are the heteropolymer containing a string of the amino acids. Structure of the molecule means the different things in the different context. In inorganic chemistry, the structure invariably refers to as a molecular formula, for example NaCl, MgCl2, etc. The organic chemistry always write a two-dimensional view of the molecule while representing the structure of the molecule, for example the benzene, naphthalene, etc. The physicist conjured up the three-dimensional view of the molecular structure, while the biologists describe the protein structure at a 4 levels. The sequence of the amino acid, that is the positional information of the protein, which is the first amino acid, which is the second, and so on, is called as a primary structure of the protein. A protein is imagined as a line, the left end represent by the first amino acid and the right end represent the last amino acid. The first amino acid is also called as an N-terminal amino acid, and the last amino acid is called as a C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of the helix, similar to the resolving staircase. Of course, the only some portion of the protein thread are arranged in the form of the helix. In proteins, only right-handed helix are observed. Other regions of the protein thread are folded into other forms in what is called as a secondary structures. In addition, a long protein chain is also folded upon itself, like the hollow woolen ball, giving rise to the tertiary structure. This gives us a three-dimensional view of the protein. Tertiary structure is absolutely necessary for the many biological activities of the protein. Some proteins are an assembly of the more than the one polypeptide or a subunits. The manner in which these individual folded a polypeptides or a subunit are arranged with respect to the each other, for example the linear string of the sphere or sphere arranged upon each other in the form of a cube or a plate, etc., is a architecture of the protein, otherwise called as a quaternary structure of the protein. Adult human hemoglobin consists of the 4 subunits. The 2 of these are the identical of each other, and hence the 2 subunits of the alpha type and the 2 subunits of the beta type together constitute a human hemoglobin, that is the HB. 9.8 The nature of the bond linking monomers in the polymer. In polypeptide or a protein, amino acids are linked by the peptide bond, which is formed when a carboxyl, that is the COOH group of the one amino acid, reacts with the amino group, that is the NH2 group of the next amino acid. With the elimination of the water moiety, the process is called as a dehydration. In polysaccharide, the individual monosaccharides are linked by the glycosidic bond. This bond is also formed by the dehydration. This bond is formed between the two carbon atoms of the two adjacent monosaccharides. In nucleic acid, the phosphate moiety links the 3' carbon of the one sugar of the one nucleotide to the 5' carbon of the sugar of the succeeding nucleotides. The bond between the phosphate and the hydroxyl group of the sugar is an ester bond. As there is one such ester bond on either side, it is called as a phosphodiester bond. Nucleic acid exhibit a wide variety of the secondary structures. For example, the one of the secondary structure exhibited by the DNA is a famous the Watson-Crick model. This model says that the DNA exists as a double helix. The two strands of the polynucleotide are the antiparallel, that is run in the opposite directions, and the backbone is formed by the sugar phosphate or the sugar chain. The nitrogen bases are the projected more or less perpendicular to this backbone but a face inside. Adenine and the guanine of the one strand compulsorily the base pair of the thymine and the cytosine respectively on the other side. There are the two types of the hydrogen bonds between the adenine and the thymine, and the 3 hydrogen bonds between the guanine and the cytosine. Each strand appear like the helical staircase. Each step of the ascent is represented by the pair of the bases. At each step of the ascent, a strand turns the 360 degree. One full turn of the helical strands would involve the 10 steps and the 10 base pairs. Attempt a drawing a line diagram. A pitch would be the 34 angstrom units. A rise per base pair would be the 3.4 angstrom unit. This form of the DNA with the above mentioned silent features is called as a bDNA. In higher classes, you will be told that there are the more than the dozen forms of the DNA named after the English alphabet with a unique structural features. Now the 9.9, the dynamic states of the body constitute the concept of the metabolism. What we have learned till now is that the living organism, be it be a simple bacterial cell like the protozoans, a plant or animals, contain thousands of organic compounds. These compounds or biomolecules are present in a certain concentration expressed as a moles per cell or moles per liter, etc. One of the greatest discoveries were made was the observation that all these biomolecules have the turnover. This means that they are the constantly being changed into the some other biomolecules and also made from the some other biomolecules. This breaking and the making is through the chemical reaction constantly occurring in the living organism. Together, all these chemical reactions are called as a metabolism. Each of the metabolic reaction result in the transformation of the biomolecule. A few examples for such a metabolic transformation are removal of the CO2 from the amino acid making and the amino acid into the amine, removal of the amino group in the nucleotide bases, hydrolysis of the glycosidic bond in the disaccharide, etc. We can list the 10 and the 1000 of such examples. Majority of these metabolic reactions do not occur in the isolated but are always linked to the some other reactions. In other words, the metabolites are the converted into each other in the series of the link reaction called as a metabolic pathways. These metabolic pathways are similar to the automobile traffic in the cities. These pathways are either linear or circular. These pathways are crisscross each other, and that is there are the traffic junction. Flow of the metabolites through the metabolic pathways has a definite rate and the direction like the automobile traffic. These metabolites flow is called as a dynamic state of the body constituent. What is the most important is that this interlinking metabolic traffic is a very smooth and without a single reported mishap for the healthy conditions. Another feature of these metabolic reaction is that every chemical reaction is a catalyzed reaction. There is no uncatalyzed metabolic conversion in the living system. Even a CO2 dissolving in the water, a physical process, is a catalyzed reaction in the living system. The catalyst which hasten the rate of the given metabolic conversion are also the protein. These proteins with a catalytic power are named as the enzyme. Now the 9.10 is a metabolic basis for the living. Metabolic pathways can lead to the more complex structure from the simpler structure. For example, the acetic acid becomes the cholesterol, or leads to a simpler structure from the complex structure. For example, the glucose becomes the lactic acid in our skeletal muscles. The former case are called as a biosynthetic pathway or anabolic pathways. The latter constituent degradation and hence are called as a catabolic pathways. Anabolic pathways are expected to consume the energy. Assembly of the protein from the amino acid requires the energy input. On the other hand, the catabolic pathway leads to the release of the energy. For example, when the glucose is degraded to the lactic acid in our skeletal muscle, the energy is liberated. This metabolic pathway from the glucose to the lactic acid, which occurs in the 10 metabolic steps, is called as a glycolysis. Living organisms have learned to trap this energy liberated during the degradation and store it in the form of the chemical bonds. As and when needed, this bond energy is utilized by the biosynthesis, osmotic and the mechanical work that we perform. The most important form of the energy currency in the living system is the bond energy in the chemical called as adenosine triphosphate, that is the ATP. How do the living organism derive their energy? What strategy have they evolved? How do they store this energy and in what form? How do they convert this energy into work? You will study and understand all this under a sub-discipline called as a bioenergetics later in your higher classes. Now the 9.11, the living states. At this level, you must understand that the tens and the thousands of the chemical compounds in the living organism, otherwise called as a metabolites or a biomolecules, are present at a concentration characteristic of each of them. For example, the blood concentration of the glucose is a normal healthy individual is a 4.2 millimole per liter to the 6.1 millimole per liter, while that of the hormones would be the nanogram per ml. And the most important fact of the biological system is that all living organisms exist in the steady state characteristic by the concentration of each of these biomolecules. These biomolecules are in a metabolic flux. Any chemical or a physical process moves spontaneously to the equilibrium. The steady state is a non-equilibrium state. One should remember from the physics that system at equilibrium cannot perform the work. As a living organism work continuously, they cannot afford to reach the equilibrium. Hence, the living state is a non-equilibrium steady state to be able to perform the work. Living process is a constant effort to prevent the falling into the equilibrium. This is achieved by the energy input. Metabolism provides the mechanism for the production of the energy. Hence, the living state and the metabolism are the synonymous. Without metabolism, there cannot be the living state is maintained. Now, the 9.12 is the enzymes. Almost all enzymes are the proteins. There are some nucleic acids that behave like the enzymes. These are called as the ribozymes. One can depict the enzyme by the line diagram. An enzyme like the protein has the primary structure, that is the amino acid sequences of the protein. An enzyme like any protein has the secondary and the tertiary structure. When you look the tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence the many cervix or the pockets are made. One such a pocket is the active site. An active site of the enzyme is the cervix or a pocket into which the substrate gets fits. Thus the enzymes, through their active site, catalyze the reaction at a high rate. The enzyme catalysis different from the inorganic catalysis in the many ways, but one major difference need mention. Inorganic catalysts work efficiently at a higher temperature and the high pressure, while the enzymes get damaged at a high temperature and says above the 40 degrees centigrade. However, the enzyme isolated from the organism who normally live under the extreme high temperature, for example, the hot vents and the sulfur springs, are the stable and they retain their catalytic power even at a high temperature up to the 80 to the 90 degrees centigrade. The thermally stable. Thermal stability is thus an important quality of such enzyme isolated from the thermophilic organisms. Now the 9.12.1, that is the chemical reactions. How do we understand these enzymes? Let us first understand the chemical reaction. The chemical compound undergo the two types of the changes. The physical change, simply referred to as the change in the shape without breaking the bonds. This is the physical process. Another physical process is the change in the state of the matter, when the ice crystal into the water or when the water becomes the vapors. There are also physical processes. However, when bonds are broken and new bonds are formed during the transformation, this will called as a chemical reaction. For example, the BaOH2 plus H2SO4, it gives rise to the BaSO4 plus 2H2O. Is an the inorganic chemical reaction. Similarly, the hydrolysis of the starch into the glucose is an the organic chemical reaction. The rate of the physical or the chemical process refers to as the amount of the product formed per unit time. It can be expressed as a rate is equal to delta P divided by delta T. The rate can also called as a velocity if the direction is specified. The rate of the physical and the chemical process are influenced by the temperature among the other factors. A general rule of the thumb is that the rate doubles or decreases by the half of the every 10 degrees centigrade change in their direction. The catalyzed reaction proceeds at a rate vastly higher than that of the uncatalyzed one. When the enzyme catalyzed the reaction are observed, the rate would be the vastly higher than the same but uncatalyzed reaction. For example, the CO2, that is the carbon dioxide plus the water, it in presence of the carbonic anhydrase, it gives rise to the carbonic acid, that is the H2CO3. In absence of any enzyme, this reaction is very slow, with about the 200 molecules of the H2CO3, that is the carbonic acid, becoming form an hour. However, by using the enzyme present within the cytoplasm is called as a carbonic anhydrase, the reaction speed dramatically with about the 6 lakhs molecules being formed every second. The enzyme has accelerate the reactions rate about the 10 million times. The power of enzyme is incredible indeed. There are the thousands of types of the enzymes, each catalyzes the unique chemical or the metabolic reactions. A multi-step chemical reactions, when each of step is catalyzed by the same enzyme complex or a different enzymes, it's called as a metabolic pathways. For example, the glucose is converted to the 2-pyruvic acid is actually a metabolic pathway in which glucose becomes the pyruvic acid through the 10 different enzymes catalyze metabolic reactions. When you study the respiration in the chapter number 14, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or the two additional reaction give rise to the variety of the metabolic end products. In our skeletal muscles, under the anaerobic conditions, the lactic acid is formed. Under normal aerobic condition, the pyruvic acid is formed. In yeast, during the fermentation, the same pathway leads to the production of the ethanol, that is the alcohol. Hence, in the different condition, different products are possible. Now, the 9.12.2 is how do enzymes bring about such a high rate of the chemical conversion. To understand this, we should study the enzyme a little more. We have already understood the idea of the active site. A chemical or the metabolic conversion refers to the reaction. The chemical which is converted into the product is called as a substrate. Hence, the enzyme, that is the protein with the three-dimensional structure including and the active site, convert a substrate into the product. Symbolically, this can be depicted as a substrate convert to the product. It is now understood that the substrate, that is the S, has to bind the enzyme at its active site within a given cleft or a pocket. The substrate has to diffuse towards the active site. There is thus and the obligate formation of the enzyme substrate complex is strand for the enzyme, and this complex formation is a transient phenomenon. During the state where the substrate is bound to the enzyme's active site, a new structure of the substrate called as a transition state structure is formed. Very soon after the expected bond breaking or making is completed, a product is released from the active site. In other words, the structure of the substrate get transformed into the structure of the products. The pathway of this transformation must go through the so-called as a transition state structure. There could be the many more altered structure state between the stable substrate and the product. Implicit in this statement is a fact that all other intermediate structural statements are unstable. Stability is something related to the energy status of the molecule or the structures, and hence we look at this pictorially through a graph. It looks like this something as in a figure. The y-axis represent the potential energy content and the x-axis represent the progression of the structural transformation or states through the transition state. We would notice two things: the energy level differences between the substrate and the product. If the product is at a lower level than the substrate, the reaction is exothermic, when the need not supply energy by heating in order to form the product. However, whether it is an the exothermic or the spontaneous reaction or an the endothermic or the energy requiring reaction, AS, that is the substrate, has to go through the much higher energy state or the transition state. The differences in the average energy content or a substrate from that of this transition state is called as the activation energy. Enzymes eventually brings down this energy barrier, making the transition of the substrate to the product more easy. Now the 9.12.3 is the nature of the enzyme action. Each enzyme, that is the enzyme, has a substrate, that is the S, binding site in its molecules, so that the highly reactive enzyme or the substrate complex is produced. This complex is a short-lived and dissociated into its product, that is the P, and then unchanged enzyme, which is intermediate formation of the enzyme product complex, that is the EP. The formation of the enzyme substrate complex is essential for the catalysis. The catalytic cycle of the enzyme action can be described in the following step. The first is the substrate binds to the active site of the enzyme, fitting into the active site. Second, the binding site of the substrate includes the enzyme to alter its shape, fitting more tightly around the substrate. 3. The active site of the enzyme, now in close proximity of the substrate breaks the chemical bond of the substrate and the new enzyme product complex is formed. Fourth is the enzyme release the product of the reaction and the free enzyme is ready to bind to the another molecule of the substrate and run through the catalytic cycle once again. Now the 9.12.4 is the factors affecting the enzyme activity. The activity of the enzyme can be affected by the change in the condition which can alter the tertiary structure of the protein. This includes the temperature, pH, change in the substrate concentration or the binding of the specific chemical that regulate its activity. The temperature and the pH. Enzymes generally function in the narrow range of the temperature and the pH. Each enzyme shows its highest activity at a particular temperature and the pH called as the optimum temperature or optimum pH. Activity declines both below and above the optimum values. The low temperature preserves the enzyme in the temporarily inactive state, whereas the high temperature destroys the enzymatic activity because the proteins are denatured by the heat. Now the concentration of the substrate. With the increase in the substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, that is the Vmax, which is not exceeded by any further rise in the concentration of the substrate. This is because of the enzyme molecule are fewer than the substrate molecule, and after saturation of these molecules there are no free enzyme molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of the specific chemicals that binds to the enzyme. When the binding of the chemical shut off enzyme activity, the process is called as an inhibition and the chemicals is called as an inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibit the activity of the enzyme, it is known as a competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result the enzyme action declines. For example, inhibition of the succinic dehydrogenase by the malonate, which closely resembles the substrate succinate in the structure. Such a competitive inhibitor are often used in the control of the bacterial pathogens. Now the 9.12.5 is the classification and the nomenclature of the enzymes. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into the different groups based on the type of the reaction and the catalyze. The enzymes are divided into the 6 classes, each with the 4 to the 13 subclasses and the name according to their 4-digit numbers. The first is the oxidoreductase, or it is also known as a dehydrogenase, the enzyme which catalyzes the oxidoreduction between the two substrate, that is S and the S dash. For example, substrate reduced plus substrate oxidized, it gives the substrate oxidized plus substrate reduced. Now the transferase, the enzyme catalyzes the transfer of the group, that is the G, other than the hydrogen between the pair of the substrate and the substrate, that is the S and the S dash. Now the next is the hydrolase, the enzyme catalyzing the hydrolysis of the ester, ether, peptide, glycosidic, carbon-carbon, carbon halide or the phosphate and nitrogen bonds. Now the lyase, the enzyme that catalyzes the removal of the group from the substrates by the mechanism other than the hydrolysis, leaving the double bond. Isomerase includes all enzyme catalyzing the interconversion of the optical, geometric or the positional isomers. And last is a ligase. The enzyme catalyzes the linking together of the two compounds. For example, the enzymes which catalyze the joining of the carbon-oxygen, carbon-sulfur, carbon-nitrogen and phosphate and oxygen etc. bond. Now the 9.12.6 is a cofactor. Enzymes are the compound of the one or several polypeptide chains However, there are the number of cases in which the non-protein constituent called as a cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called as the apoenzymes. And three kinds of the cofactor may be identified: the prosthetic group, coenzyme, and the metal ions. Prosthetic groups are the organic compound and are distinguished from the other cofactor in that they are tightly bound to the apoenzymes. For example, in a peroxidase and catalase, which catalyze the breakdown of the hydrogen peroxide to the water and the oxygen, heme in the prosthetic group, and it is the part of the active site of the enzyme. Coenzyme are also the organic compound, but their association with the apoenzyme is only transient, usually occurring during the course of the catalysis. Furthermore, the coenzymes serve as a cofactor in the number of the different enzymes catalyze the reaction. The essential chemical component of the many coenzymes are the vitamins. For example, the coenzyme nicotinamide adenine dinucleotide, that is the NAD, and NADP contains the vitamin niacin. A number of enzymes require the metal ions for their activity, which form the coordination bond with the side chains at the active site, and at the same time form a one or the more coordination bonds with the substrate. For example, the zinc is a cofactor for the proteolytic enzyme carboxypeptidase. The catalytic activity is lost when a cofactor is removed from the enzyme, which testified that they play a crucial role in the catalytic activity of the enzyme. Now, the summary of the chapter of the biomolecules. Although there are the bewildering diversity of the living organism, their chemical composition and the metabolic reaction appear to be the remarkably similar. The elemental composition of the living tissue and the non-living matter appear also to be the similar when analysis is qualitatively. However, a closer examination reveals that the relative abundance of the carbon, hydrogen, and oxygen is higher in the living system when compared to the inanimate matter. The most abundant chemical in a living organism is the water. There are the thousands of the small molecular weight, that is the less than the 1000 Dalton biomolecules. A amino acid, monosaccharide, and the disaccharide sugars, fatty acid, glycerol, nucleotides, nucleosides, and the nitrogen bases are the sum of the organic compounds seen in the living organism. There are the 20 types of the amino acid and the 5 types of the nucleotides. The fats and oils are the glycerides, in which the fatty acid are esterified in the glycerol. The phospholipid contains in addition a phosphorylated nitrogenous compound. Only 3 types of the macromolecules, that is the protein, nucleic acid, and the polysaccharide are found in the living system. Lipids because of their the association with the membrane separate in the macromolecular fraction. Biomolecules are the polymers and they are made up of the building blocks which are the different. The proteins are the heteropolymers made of the amino acids. The nucleic acid like the RNA and the DNA are composed of the nucleotides. Biomacromolecules have the hierarchy of the structures like the primary, secondary, tertiary, and the quaternary. Nucleic acids serve as a genetic material. Polysaccharides are the component of the cell wall in the plants, fungi, and also of the exoskeleton of the arthropods. They also are the storage form of the energy, for example, the starch and the glycogen. The proteins serve as a variety of the cellular functions, and many of them are the enzymes. Some are the antibodies, some are the receptor, and some are the hormones, and some other are the structural proteins. Collagen is the most abundant protein in the animal world, and the ribulose bisphosphate or carboxylase oxygenase, that is the rubisco, is the most abundant protein in the whole of the biosphere. Enzymes are the proteins which catalyze the biochemical reactions in the cell. Ribozymes are the nucleic acid with a catalytic power. Proteinaceous enzymes exhibit a substrate specificity, require the optimum temperature and the pH for the maximum activity. They are denatured at a high temperature. Enzymes lower the activation energy of the reaction and enhance greatly the rate of the reaction. Nucleic acid carry the hereditary information and are passes from the parental generation to the progeny. Thank you so much.